Hola a todos, buenas tardes. Bienvenidos a esta conferencia que será impartida por el doctor Jean-François Coté, quien es investigador de la Unidad de Investigación de Organización Citoesquelética y Migración Celular del Instituto de Investigación Clínica de Montreal. Agradecemos a todos ustedes su participación y su asistencia a esta conferencia, particularmente un agradecimiento a la Dirección de Investigación y de Docencia del Instituto por todas las facilidades que nos proporcionaron para la organización y la realización de este evento. Entonces, permítanme presentar al doctor en inglés, dado que la conferencia va a ser en inglés. Trataré de hacer mi mejor esfuerzo, lo prometo. Entonces, lo presentaré. Hello, everyone. Thank you very much for attending this conference entitled On the Move and Hiding, Dissecting the Breast Cancer Journey from Dissemination to Dormancy, given by Dr. Jean-François Coté, Director of the Cytoskeletal Organization and Cell Migration Research Unit at the Montreal Clinical Research Institute. He is a research professor in the Department of Medicine at the University of Montreal and adjunct professor in the Department of Anatomy and Cell Biology in the McGill University. Dr. Jean-François has published more than 100 scientific articles, many of them in prestigious journals, including Nature Communication, Nature Cell Biology, Cancer Cell, Cancer Research, and Proceedings of National Academy of Sciences, among others. His work focuses primarily on elucidating the molecular mechanisms underlying breast cancer cell migration, invasion, metastasis, and dormancy. Welcome, Doctor, to the National Cancer Institute. It is a pleasure to have you here. The stage is yours.
Gracias for the invitation. I'm very happy to be here and present our work today. And please ask a question, English, French, and translation in Spanish. Omar is going to translate. So today I'm going to tell you, I prepared a seminar with a bigger story and a smaller story that I want to present at the end on the GTPAs. And I'll give a little bit of introduction on our interest in metastasis and the growing interest in the tumor dormancy. So as you know, the breast cancer is a very complex disease. Breast cancer is not one disease, but is in fact many subtypes of disease. And each of these subtypes have different behaviors, prognosis, and response to treatments. Also, as I'm sure you know, the progression to the metastatic disease is really what is the major threat to the patient with breast cancer. And still to this day, we have a lot of work to do to be able to better understand this. I'm going to talk today about disseminated tumor cell and their capacity to undergo dormancy. So what are these cells? So these cells are the cells that can disseminate from the primary tumors and circulate in the body and then enter secondary organs where the cells will enter a state of dormancy until they can reactivate, reawake, and form metastatic relapses later after the patient treatment. Okay. So what are the limitations and why are we interested in this problematic? So we are really interested to provide basic knowledge such that we can better understand metastasis and one day be able to cure metastatic disease. And as the lab has progressed over the years, we've become really interested by understanding how they migrate, invade. But this has become a little bit blurry in the recent years when we realized that a lot of the cells have actually disseminated early in tumorigenesis. So, and also by the fact that we're pretty much unable to detect these single or these microcluster of cells that have disseminated into other organs. So as such, the naive view of thinking that we're going to block cell migration, we're going to block metastasis, I think is not a model that's going to hold true over a long time and being able to treat metastatic disease. So we need to deeper understand this. Okay. So this is, again, a bit of an introduction to put us all on the same page. So when the mammary breast tumors form, as you know, these mammary breast tumors are not only the epithelial cancer cells, but they form like a tumor microenvironment where the tumor cells cohabit with other cell types, such as fibroblasts, neutrophil, macrophages, and the blood vessels. And collectively, this microenvironment is important for the progression of the cancer, including to promote or fuel the ability of some of these cells to undergo some molecular changes, acquire more aggressive behaviors, and undergo the step of intravasation, which is leaving the primary tumor, entering the circulation, reaching secondary organs, such as the lungs. And then once they're in the lung, they have different fates. So some of these cells will be able to form overt metastasis right away. So some of patients are seen with metastasis already. But in many of the breast cancer subtype, these cells also choose the fate to undergo dormancy or remain as a microcluster of dormant cells. Okay. So this is what I was trying to tell you a little bit in the introduction is when the tumor is very small, okay, it is clinically undetectable. And as the tumor grows bigger and it becomes detectable, what we have learned, and this is what I learned when I was in school, is that it's when we have big tumors like this that there will be a lot of cell shedding, a lot of disseminated tumor cells that will be the source of the metastatic lesions. But now we know actually from very more recent literature in the last 10 years that the window of opportunity for cell to disseminate from the primary tumor can start as soon as there are very small tumors. And as such, since the tumors are not yet detectable, but they're allowed to disseminate, it kind of really confused the way we can now address metastasis. But what happened is that after surgery and therapy, then the tumor become again undetectable. And then that gives another window opportunity for the cancer cells that have disseminated to reactivate themselves. So if we're thinking about the subtype of triple negative breast cancer, as I'm sure you know, these are really aggressive cancer, and usually the relapse will be from months to 5 years that you will get these clinically detectable metastases. And again, this is not a good prognosis. But for other subtype of breast cancer, like the luminal A, luminal B, and HER2 positive breast cancer, the state of dormancy can last for decades. So between 5 to 20 years, you can have these dormant cells that are going to stay there before they decide to reactivate. So our new way of thinking, and this is what we try to put in the paper of Cancer Cell we published recently, is the idea that this gives now also new windows of opportunity to identify the vulnerabilities of these disseminated tumor cells that have not yet formed relapses and try to eliminate these dormant tumor cells. So that's what the project is about. Yeah, and I think it's also important that we think deeply and realize that, in fact, there are the primary cells that have disseminated prior to the treatments, and then there's also the disseminated cells that have also undergone treatment, and they've also acquired different resistant behavior. So at the end of the day, in this context, it's a mixture of these two cell populations, and these two populations may have different vulnerabilities. So you might not be able to eliminate them with a single approach. Hence the need to deeply understand the metastatic behavior of these cells. Okay. So this is again the same thing. It's more graphical, but it's to illustrate that even when the breast cancer patient has a bit of a small breast cancer, there can already be single cells or micro cluster of cells disseminated in the body at this time, even though the only detectable thing is the primary tumor. And the patients will undergo the treatment that's appropriate for them, eliminate the primary tumors, and these residual cells and secondary organ will have a window of opportunity to relapse over time. And it's important, the question here is what is the molecular basis of this reawakening, and what are the vulnerabilities of these cells? Okay. It's a field of research that's really going under expansion. It's really interesting, and I give an example of a paper that was recently published that I find fascinating, that explains why the luminal A type of breast cancer can stay dormant for such a long time. In this paper by France Turel and co-authors from France, they beautifully demonstrated, in fact, that the signaling cue that allows these luminal A cells to reactivate a long time after the tumor is because the lung of the animal is aging. So when the mice are aging, there's more collagen, more attraction of cells like macrophages that secrete ligand, such as this ligand here. And then this now creates a new cocktail that reactivates these cells. So for the luminal A, I think this is a paper that was really important, and that's going to be a lot of research that's going to derive for this. So the aging of the microenvironment at secondary site will be a key factor to reactivate these tumor cells. And it makes sense with the fact that these women afflicted with luminal A breast cancer can, be disease-free for decades before there's relapse. But for other type of cancer, like HER2 positive or luminal A, luminal B, much less is understood. Okay. And this is a picture that I took from the review we published a couple of years ago, where we really did an extensive review of the literature. So if any one of you is interested in dormancy, we've went to the really old paper all the way to the modern paper and did an in-depth review for the thesis of Islam. And these are some of the examples. So the programming of the primary tumor cells to enter the secondary organ as dormant cells can start from the primary tumor itself. So there's already crosstalk going on among all the cell population in the primary organ, and that can prime the cells to acquire dormant fate once they reach secondary organ. And basically across this journey of invasion and migration to secondary site, there's all type of signaling pathways that have been elucidated over the last decade. I would say the molecular biology of this is fairly recent. And I think we only touched the surface of all the complex signaling that's ongoing. But here it gives some example. Okay, so we wanted to tackle the problem using a model of triple negative breast cancer. And we stumbled across these cell lines. So that have very interesting properties. So some of you may have heard this. The very popular cell line is a 41 cell. It's a mouse mammary cancer cell that's very widely used in the literature. But if you go back to the original literature, all these 4 cell lines here were isolated as different clones of a spontaneous tumor from a bald mice. And over the years, people have characterized this different clone for their different behavior in tumor growth, metastasis, and all this. And as this was summarized here by Filippo Giancotti, you can see that one of the subclone here was able to take as a primary tumor in mice, but didn't progress more in the aggressivity. And this other line here would make a tumor growth and colonize the lymph node, but not much more. And what's interesting, we really like these 2 models here because they have pretty much the same behavior, except that the 41, once they reach the lungs, they make full-blown metastasis right away. They're really aggressive. And this very similar subclone will go all the way, but the cells will remain as dormant cell in secondary organs. So there's been a lot of studies using these cells, and we thought maybe we can apply more modern approaches like functional genomics to these cells and try to go a bit deeper in the molecular pathways that could be involved in these cells. So as a summary from this, the 4007 and 41 have been propagated in cell culture for more than 50 years, yet they retain these differential metastatic properties when we bring them in an in vivo context. So this was the rationale for us to apply CRISPR-based genetics on them and try to see if we can have some different signaling wiring in these 2 cell model. And if we do, then can we try to leverage some of these changes in signaling for therapeutic properties? Okay. So this is what we did with the project. Right. Basically, we took these cells, performed genome-wide CRISPR screen loss of function, identified the genes that are required for proliferation survival, and then did classical bioinformatics to try to identify the different signaling pathways. I'm going to take you through some of these data that were just published last week in Cancer Research. So if you want to look at it. And this was a really cool project. This was led by an MD student that came, MD from Egypt that wanted to do a PhD, Islam El-Kholi. So he came to Montreal in my lab, wanting to learn about molecular stuff. And over his PhD, he really accomplished a lot of nice work. So he's done 90% of what I'm going to tell you about. And we leverage great collaboration. So CRISPR experts in Toronto, breast cancer expert in Montreal, dormancy expert in New York, and Washington and Florida, so all over the US. So it was a really fun project to work with these folks. So what we did at the beginning is the 414007 have been used for a long time, but we didn't know much about really what type of subtypes they mimic. So we downloaded a lab data set and compared the gene expression profile of 414007 to all the human cell lines we could get our hands on. And basically, the defining features were that these cells look like basal B, breast cancer, luminal basal B, breast cancer, so triple negative like, but the basal B subtype. And through some data I'm not going to show you today, but we also did RNA sequencing, whole genome sequencing, and the CRISPR to launch that project on that. Okay. So that was the classical experiment, the CRISPR experiment that we did. So we took both cell lines, infect them with a big genome-wide library, grow them for several passages, and at the end extract the guide RNA, compare them to the beginning. And we were able to identify, as one would expect, the essential genes that are common to both cell lines, but also be able to identify unique essential gene in the 41 cells and in the 4007 cells. And this is also a simplified view to try to put some of the proteins that we were interested in. So this is where we become biased in our data. So Islam looked at all these differentially essential genes and decided to focus on the PI3 kinase families. So what this tells us here in the 41 cell, so the one that make metastasis really quickly, they're dependent on classical PI3 kinase signaling. But if you look at the dormant cell, they add several subunits of the pathways of mTORC1, including 2 subunits of the class 3 PI3 kinase. And I'm going to go a little bit more in details in that. Okay. Yeah, so that's visually how it looked like. As I'm sure many of you know, receptor tyrosine kinase signaling often activate class 3 PI3 kinase class 1. And this is a pathway that's known to be activated in many cancer, including 40% of breast cancer, activates AKT, crosstalks with the mTORC1 pathway, whereas the PI3 kinase class 3 is more involved in processes like autophagy, response, sensing amino acid and doing molecular response to amino acid, and a primary signal to the mTORC1 pathway. So this is where we started and try to dive deeper in these proteins. So if we look at the 41 cell, the metastatic cells, these are all the subunits that came out in the screen. So these were lost, then the cells were depleted from the culture. And when these were lost, then we had more of these guide RNA in the culture. So several components of the pathway were found in the CRISPR screen. And then we wanted to confirm these data in a different approach to confirm the screening data. So what we did here is a cell competition assay where we took the 41 cells that stably express the Cas9 protein and then infect them with a guide RNA that's going to target the ROSA locus with a mCherry that's also expressed in these cells. So these cells would be red. And in the other cell population, we would express guide RNAs against the classical PI3 kinase, and then mix these cells, put them in culture, and see if there's going to be a winning population. So the prediction would be that losing the PI3 kinase will lead to less of the green cells. And this is what we observed. So this is what it looks like at the microscope. Islam took pictures every day and quantified the number of red and green cells. And what it looks like at the end of the day is that three guides RNA for the PI3 kinase gave the same phenotype of dropping out these cells in culture compared to the control conditions. So yes, we could confirm the CRISPR screening that this PI3 kinase is essential in the 41 cells. I'm not showing you the data. We did the same experiment in 41, and I'm going to show you a little graph after. And that's for the 4007 cells. And these are the subunits that came out in the screen. So all these ones, when they were lost, the cells would drop out of culture after several days in culture. So this is why we thought that this would be an interesting pathway to go deeper. Okay. Again, we did the same experiment. So we took the 4007 this time, and used guides RNA against the two subunits of the class 3 PI3 kinase, and showed that these cells were quite dependent on that PI3 kinase. And if we look if it's essential in both cell lines or not, again, it confirms the CRISPR screen. So the 41 cells were not so much dependent on the class 3 PI3 kinase, but the 4007 cells are dependent on it. So we were quite satisfied with these data, and then we wanted to know a bit more about the signaling pathway that's at play. Okay, we could also confirm it with the pharmacological inhibitor. So if we use classical PI3 kinase inhibitor, the BYL inhibitor, you can see that the 4007s are not susceptible, but the 41 cells are susceptible. Again, it confirms the genetics. And then if we look at the signaling pathway, what we found that was interesting is that it was consistent with the genetic observation that there's more PI3 kinase signaling by looking at phosphoryl AKT in the 41 cells in comparison to the 4007 cells. But if we look at the mTORC1 activity by looking at phospho-S6 kinase, you can see that if you do the ratio of the phospho-S6 to total S6, then there's a lot more activity in the 4007 cells. And it's plotted like this here. So the PI3 kinase activity classical is lower in the dormant cell, but the mTORC1 activity is higher in the dormant cell. So that's really the foundation of what we wanted to dive deeper in. Okay. So I'm sorry that some slides are going to be a little bit off because we transfer to another computer, but it's okay. So I told you in the introduction that there's a lot of crosstalk between receptor tyrosine kinase, amino acid sensing, etc. So what I want to introduce you here is some of the concept of how this mTORC1 complex is activated. So as you may know or not, the mTORC1 complex is sitting on the lysosome membrane inside the cell. And when it's sensing amino acid, if you want to get robust activation of the mTORC1 pathway, you need to move these lysosomes from inside the cells closer to the plasma membrane. Okay? And I'm going to show you some of these images after. But in terms of signaling, it's again consistent. So if we look in more details in these cells, as I told you, there's more mTORC1 activity in the dormant cells compared to the metastatic cells. If we remove the serum, so if we remove this part, we get the classical response that we lose the signal here. But in fact, in the 4007, we don't get this decrease. So this is not going through this pathway here, so it's sustained. But if we remove amino acid, then you can see we completely lose the phosphorylation. And if we put amino acid back, we can restore some activity here. But in the dormant cell model, if we remove amino acid, we get a very sharp drop that can be very robustly reactivated with the amino acid. Okay, so these are the signaling experiments that told us maybe we should look at the cell biology of where these complexes are localized on lysosomal membrane, and if these cells integrate better at the plasma membrane. Okay. And these are the seminal discoveries published by other teams that identified that. So in 2011, there's a group that was working on a GTPase called ARL8 that showed that it can move lysosomes. So if you deplete ARL8, the lysosome goes around the nucleus. But if you overexpress AL8, then you push the lysosome at the cell periphery. So this was a very important paper to open up this field of research. And then consistent with the model I told you about, this other paper demonstrated that the mTORC1 complex that's localized on the lysosome needs to be transported on microtubule closer to the plasma membrane, where you get optimal response from growth factor and amino acid to maximally activate mTORC1 signaling. So that gave us the idea that maybe in our cell model, what we're dealing with is different localization of these complexes. Okay. And that's just a graphic way of representing it. So this is what it looks like. So in our dormant cell model, you can see that the lysosomes are really pushed at the periphery of our dormant cell model. But if we compare to the 41 cell, the metastatic model, you can see that the lysosome are more localized at the perinuclear region. So the genetic, the signaling, and then the cell biology is very consistent with the differential signaling of these pathways. Then the next question we wanted to know is this, very clear different localization, is it dependent on the activity of the class 3 PI3 kinase? And the answer is yes. If we use the inhibitor VPS34, which is an inhibitor of the class 3 PI3 kinase, you can see that in these 4007 cell, untreated, they're nicely localizing the lysosome at the cell periphery. But if we put the inhibitor, then they move to the peripheral region. And in the 41 cell, they're already there, but they get even tighter around the perinuclear region. So this is quite exciting finding for us. And then we wanted to know, can any of these findings translate into the in vivo setting? So if the genetic and the biochemistry and cell biology we did is true, we don't think that the class 3 PI3 kinase will be very important in the aggressive cell lines, the 41 cells. But of course, to be comparative, we studied both models, even in the in vivo context. So here in this experiment, we injected the 41 cells in mammary fat pad of the mice, let the tumors grow. And because they grew to really big size, we resected the tumor and then treated with the VPS34 inhibitor and quantified primary tumors and metastasis. Okay, and basically there's no difference. So if you look at the number of metastases, it's the same. The total tumor growth, I don't know if I show it here. I didn't show it, but the total primary mass is unaffected. And if we look at how the metastatic lesion looks in the lung, they look the same. Okay, so that was exciting, but we wanted to know what will happen with the dormant cell model. So to do this experiment, it's a little bit more tricky because these cells will not form metastasis once they reach the lung. They will stay as single cells. So we need to rely on other strategies to be able to quantify the number of dormant cells in the lungs. So to do that, what we did in that experiment is we injected 200,000 cells in a mammary fat pad, waited 3 weeks that they form a relatively big tumor, and then treated these mice with the VPS34 inhibitor or control vehicle condition. Okay. And then what we did to be able to quantify the number of dormant cells in the lungs is we exploited the fact that these cells have been made resistant to thioguanine. So what we did is we isolated the lung from these animals at that stage, digested the lung with collagenase displase, and plated the cells in many dishes, and then select them under thioguanine for 7 days. And what grew out of these cultures are colonies that originate from the 4007 cell. So this is classically done by other labs. We just adapted the technique here. And this is where I think we got interesting results. So if we quantify the primary tumor burden, it was the same in both animals. So the VPS34 inhibitor does not affect the growth of the tumors. But if we quantify the metastatic lesion by counting the number of 4007 cells, then there's quite a significant decrease in these cells that survive in the secondary organs. Okay, so this is how the paper was submitted to the journal. And then one of the reviewers said, all the work is done on 4007, 41 cells. Please find another cell model and redo all the project. So So this is why we had so many collaborators who came and helped us. We collaborated on this one with James Alvarez for Washington, who I think you are going to like also this cell model. So James's lab is working on HER2 model of breast cancer. And he can regulate the expression of the HER2 oncogene with the doxycycline molecule. So what we did in that experiment with James is that we grew tumor in these mice. And after we had mice there, we separated them in the mice with primary tumors. We made 2 models of them. One that we kept on ducts, so they had tumors. And we made cell line out of these tumors. And another portion of the mice were removing the doxycycline, shutting down the HER2 oncogene, so the tumors would disappear. But then 100 days later, then there would be relapses or primary tumor relapses that would come out. And then we made cell line of these relapses as well. And much like the 41, 4007 cells, we isolated these cells and did whole genome CRISPR screening on them. And this is what we found. So we found that in these cells that were originating from non-dormant models, the PI3 kinase in the CRISPR screens were not essential. But in these cells from the dormant models, then the PI3 kinase, the PI3 kinase became essential genes, very similar to what we've seen in the other model. So we think that at least in 2 models like this, these 2 subunits of PI3 kinase class 3 are good therapeutic target for this. And while this work was being published, 2 groups on ovarian cancer also called us and told us that they found this PI3 kinase in screen of dormancy for ovarian cancer. So we're quite excited that this might become an attractive target in the future. But we pushed it just a little bit more further with the HER2 model. We confirmed it with the inhibitor in culture. So if we put the inhibitor, the dormant model died more than the control. And if we did the dormancy experiment in vitro, so put the cells in culture with ducts or without ducts, and then treat them with the VPS inhibitor, we could see that it's the dormant cells that if we put the inhibitor would start to undergo apoptosis. So this is the conclusion from that study, that from a lot of deep characterization of a cell model of dormancy and aggressive metastatic behavior, we were able to focus on 2 signaling pathways to explain these behaviors. Most interestingly, in that study, we found the regulation of the lysosome appears to be essential to promote the survival of these dormant cells. And once these cells seeds as single cells in the lung and we inhibit this pathway, then we can start to eliminate some of these dormant metastatic cells. So I think this is the first study that's published in breast cancer using a complete landscape, using like genome-wide approaches to get new candidates. What is interesting is we publish all the data set with that paper. We focus on this pathway, but anybody who are interested can look at these data, and I think there's a lot of other interesting hits that could be also characterized. I think it was really interesting, again, the positioning of the lysosome, this very fundamental cell biology experiment that lysosome localized differently. We can find a biological function in cancer here. Pharmacological inhibition of PI3 kinase was able to decrease metastatic burden in 2 models, triple negative and HER2. And then we got criticized. This is where I become a bit philosophical. The reviewers for that paper, one of the reviewers like, it's triple negative breast cancer cell lines and triple negative breast cancer in patients in clinic relapse in 5 years or less. So why do we care about dormancy in these models? I think we kind of strongly argued this fact because a lot of our collaborators and our clinician collaborators think that it would be of great benefit to a triple negative patient, even if they have early relapse, if we could decrease the number of dormant, or even if they're not fully dormant cells, as the reviewer was claiming, if we're able to find vulnerabilities in these cells in that window of opportunity before they can relapse, I think we could significantly extend the life of these triple negative breast cancer and their quality of life. So, Omar, am I okay on time? Yeah, okay. So this concludes the first part. And then I have a few experiments on another project that I thought it would be interesting to show on the GTPase protein that we've been working on. So what I'm going to tell you in that shorter story is a lot of it is based on the fact that we use proximity bioaccumulation approaches to map protein-protein interaction at high throughput in mammalian cells. And here I'm going to show you some very preliminary result on the RAS family, where we would like to discover new interactors of RAS, especially oncogenic RAS, to be a potential drug target in the future. Yeah. Some of you, I know there's some people interested in raw proteins. There's more than 150 RAS proteins in the human genome, if you didn't know. And they're divided in families, including RAN, RAB, ARS, raw proteins, and the RAS, the classical RAS oncogenic proteins, okay? But of course, as every field of research, everyone is working almost on the same proteins. So out of this full genetic tree, only a handful of these proteins have been characterized to great depth, and the other have been superficially analyzed. So in my lab, we had this idea that why we should be able now to leverage the new techniques and try to study this thing as at high throughput, okay? So these are the famous experiments that were done by Alan Hall. So he had found that RHO can form stress fiber, RAC1 can form lamellipodia, CDC42 forms filipodia. I'm sure you're all familiar with this. But all these other proteins, there's 20 of those. So RHO-B, RHO-C, RAC23, RHO-G, RHO-G, RHO-Q. These are drastically understudied in comparison to the classical protein. Okay? Like in 2000, like 10 years ago, when this technique was starting to emerge, the proximity bioaccumulation approach, we jumped on this and said that this was going to be potentially a very strong technology to identify new interacting proteins of raw protein. And I'm going to tell you why. So you're probably all familiar with this approach, the affinity purification approach, where the protein that you're interested in is fused with a etiqueta, I think you say, Spanish, etiqueta. And then you use this tag to purify the proteins and identify its interacting proteins. As I'm sure you know, the limitation of this approach is that the nice protein complex have to resist purification and washing, and some of the weak interaction won't survive this, okay? Why did we think that it would be good for GTPase to use BioID? It's because the bioaccumulation of the nearby proteins will be occurring in the live setting, in vivo in the cells, when the cells are alive. And what you do is you don't purify the protein complex, you purify the bioaccumulated proteins. So in fact, you don't need the protein to interact together to do BioID. The interaction will have taken, the labeling will have occur, and then you purify the labeled proteins. And this labeling is thought to be occurring in about 10 nanometer around the biotin ligase. So we use BRA here in this experiment. So you label a radius of about 10 nanometer. And then this is a really powerful approach because, you can easily isolate biotin labeled peptide with streptavidin, which is a very, very efficient approach. Okay. So this is what we did in that paper we published in 2020 in Nature Cell Biology. And here it's a picture showing all the known interaction. That's why I titled this the power of BioID. So we did the 20 raw proteins in these assays. So this is very easy experiment to do. It looks fancy and complicated, but it's in fact very easy. And it's amenable to high throughput. So Anil in the lab made many cell lines expressing all these 20 proteins, active state, inactive state, mutant state, and conducted 200 experiments of BioID or so. And this is just to show you that all the work that's been done in the 40 years before, we could recover most of the interaction that were published, plus a layer of new one that if you're interested, you need to read that paper. Okay. And then more recently, we decided to jump on another family, the ARF protein. This was done by Laura, amazing student in my lab in collaboration with structural biologist Matt Smith. And the ARF proteins, there's 29 of these ARF proteins. They regulate vesicular transport between organelles. And much like the Rho proteins, everybody studied ARF1, ARF6, and a few of them, but very few were studied systematically. So Laura did the same pipeline, labeled everything. And again, I'm showing you some of the new stuff. We identified ALF14 bound regulator of magnesium. We identified all the known interactor of SAR1 and blah, blah, blah. And this paper was published recently. So we became really good with that technology. We created this large data set we deposited with these papers. There's a lot of information that can be leveraged for new experiments, et cetera. And then we became very interested in the RAS protein to push approaches technology there as well. Okay, and I'm sure you know here in the Cancer Center that RAS is a major oncogene. And then when you get unbalanced regulation of RAS, so if RAS is GTP and active, you get uncontrolled growth, proliferation, and migration. The signaling pathways regulated by classical RAS protein are well established. There's thousands and thousands of papers on how it works. And in the clinic, I'm sure you also all know that pancreatic cancer, as 95% of them have mutation in KRAS as an oncogenic driver, colorectal 45, lung cancer 35. So KRAS is a major human oncogene. NRAS in 15% of AML, 15% of melanoma, and HRAS in about 10% of bladder cancer. Okay? And if you didn't know, it's very interesting also that for each of these oncogenic RAS, they come in many variants. So the activating mutation is not always the same. And there's the hypothesis that each of these different variants, even though the GTPase is active, will have preferential binding partners. So this is still at the hypothesis stage, I would say. And there's been a huge regain in interest in trying to drug KRAS with the discoveries from the Kevin Schuchat lab in San Francisco, where they were finally able to identify the first drug that can specifically target the G12C mutation in KRAS. So it's a covalent drug inhibitor that inhibits this. It's been FDA approved now, but it's showing high level of resistance over time. But I think this has completely revived the field to start to be hunting for more ways to try to target RAS protein. But that's why we got interested by that and go a bit deeper. And so these are the three oncogenic RAS. And like I said, most people work on these three, and these are much more or less studied. So again, we took a systematic approach. We did bio-ID on all of these cells. So what we did is 3 control cell lines, 8 wild types, GTPases, 35 of them in a constitutively active state, 4 different conditions where we took some of these funky mutations. And then we did all these experiments in 3 cell lines. And again, this is done by Laura. She generated over 300 bio-ID samples that are now all analyzed. And then she got these big data sets like this. So you can see these are all the interacting proteins collectively of the 35 samples. So we have some common interactors, many uniques, and all that. This is what it looks like. The proteins localize as they should in cells. And if we do the biotin staining, the cells are heavily biotin stained. And then I think what Laura did was very clever. She crossed all our interaction database with the depth map library online that identifies co-vulnerabilities. So basically gene that would be synthetic lethal with your gene of interest. And what she found for the oncogenic RAS1 is a lot of the expected players. So everybody who knows KRAS knows that it's signaled a lot through RAF1. Well, RAF1 is essential also there. But we identified this protein DOC5, which is a RAC1 activator, coming up in all these cross-referencing database. And when we go and download the data from the depth map, and we look at which genes are codependent with KRAS, so which gene that if are co-knockout with KRAS would be synthetic lethal, DOC5 ranks as the first one, even better than RAF1. Yet there are thousands of papers on this and zero on DOC5. So we think that the BioID was an approach to be able to identify an interaction that was previously undetectable by classic IPMS experiment. And now, if we look at this genetic data that speaks volume, DOC1 is a very essential gene in KRAS cancer. Now we start to understand that the KRAS might be directly binding to the DOC5. Okay. And that's another way of looking at the data and showing that the DOC5 was dependent in all the 3 cells model we look at. But for example, for some other, like the beta-catenin, it was more prominent in 2 cell lines and less in 1, etc. So there's lots of data to explore. Okay. So the question is, does DOC5 and RAC interact? So Laura made some experiments to try to test this. And she showed that indeed the KRAS can co-precipitate with DOC5. And if she does functional experiment with a DOC5 inhibitor that was generated previously by other teams, C21, and we use a lot of KRAS-dependent cell line. So in most of these cell lines, if we treat them with the DOC5 inhibitor, we abolish growth significantly. In this experiment, there's 2 abnormalities. This one, which is a constitutively active KRAS that did not respond. We don't know why. There might be another driver. And this one with wild-type RAS that also responded, which doesn't mean it could also be dependent on RAS signaling to DOC5 there. But the cell line is not mutated for RAS. Okay. And then the same thing we could confirm with the genetic approach. So this is more recent data. She used another cell line. This time she also wanted to start to include lung cancer cell lines. So if she knocked down DOC5 by siRNA, then you can see the cells grow less also. So this is a preliminary conclusion from this project. So the BioID was completed on 35 RAS proteins in 3 cell lines. We identified many new proximity interaction, especially for the understudied RAS. But we decided to focus on the classical RAS. This was not the plan, but this is what we did because we found this DOC5, and which DOC5 I discovered in my postdoc. So I was going back to my love. Yeah. So what can we do there? We're trying now to map this KRAS DOC5 interaction. I showed you some co-IP that works. But we tried a lot of experiments like using GST protein, purified protein with smaller protein fragments. Yet we have not been able to reconstitute the DOC5 interaction with RAS in purified condition. And this is because we think that it's probably the binding site is coming from many parts on the protein in the 3D structure manner. So Laura is like actively working on that with the structural biology collaborator. We're doing a lot of cell biology, so we're using approaches like trying to activate KRAS with photoactivation and see if it will recruit DOC5 at the membrane. We have a collaborator looking at patient samples, trying to use the IC51 in more in vivo experiment. And one of our collaborators at the Institute is developing an ASO therapeutic to try to see if we can put it in the mouse model. So this is what it looks like at the end. But we think this DOC5 protein is a huge protein of over 200 kilodalton, and it's bound with many other co-functioning subunits. So we think that it will be a little bit challenging to map how the RAS was binding to it. And if it would be easy, people would already find it with all the activity undergoing the RAS field for several decades. So Laura is really using structural approach for that. Yeah, and that concludes my presentation. And a word for the students, if any is interested in Montreal, scan this QR code for grad school or postdoc or whatever. This is Montreal in the summer. And if it was in the winter, it would be all like lab coats everywhere. It would be white. Yeah. And then, yeah, just I think the people, Islam did all the first project on dormancy. Laura is the star of the second project on the ARPS and the RAS. Our collaborators, our funding agency. And I want to thank you for the invitation. I'm very honored to be here and present to you, and happy to take any questions.